Hi biology, this recording is going to go over the lesson how cells make proteins. I'm going to tell you this is a long recording. There is so much information in this lesson, so please feel free to pause and come back, okay? Now the objective of the lesson is to use reasoning to connect evidence that shows the relationship between the structures and functions of DNA and proteins. But to do that, we need to understand how we go from DNA to protein, um, and then you kind of infer that there's that structure determines the function, which we'll talk about towards the end. Remember, yellow star means a quick check question is being covered. Last lesson, you learned more about DNA, how DNA is, we previously had learned about how DNA is made up of cysteine, guanine, adenine, and thymine, that those are the nucleotides. In the last lesson, you learned that that's wrapped around proteins, that creates chromatin, and that it's organized as chromosomes, and that you have that base pair rule to help copy the DNA so all your cells have the same DNA. So what we're covering today is referred to as the central dogma of genetics. So how genetics works. And the central dogma, like the main component, is that DNA gets changed into RNA, which then changes into a protein. And to add some vocab there, DNA to RNA is called transcription. And then RNA to a protein is translation. And I know the words are very similar, but think of it like this. From DNA to RNA, we're using the same language. It's still nucleotides being made, the nucleotide structure. So we're just rewriting it. It's the same language, we're just rewriting it. But then from an RNA to a protein, that's translation, right? We're translating it into a different language. So we have RNA, which is made out of nucleic acids, and we're translating it to an amino acid sequence. This whole process is called gene expression. So gene expression is when a segment of DNA, which is called a gene, is actually made into a protein. So that segment of DNA, in this picture it's keratin, gets changed into RNA, and that RNA can leave the nucleus and then be translated into a protein, and this protein does something in the body. And the sequence of amino acids is important. It determines the shape of the protein and how the protein can function. All right, so let's first review the differences between RNA and DNA that gets covered in this lesson. So RNA is ribose nucleic acid, and then DNA is deoxyribose nucleic acid. Sorry, my brain went blank. Both are made of nucleotide. The thing that's different between the two is their sugars. The sugar has, the DNA sugar has less oxygen. So if you look here, this picture, so that's why it's a deoxyribose sugar, and this is just a ribose sugar. They also have a phosphate, and then each nucleotide has a different base, and that base determines the type of nucleotide. But there are nucleotide differences between the two. So DNA has adenine, guanine, cysteine, and thymine. RNA has the first three the same, but instead of thymine, it has uracil. So uracil is going to base pair with adenine to make RNA. And that goes back to the last lesson about base pairing. So C and G always base pair, and A and T always base pair, but if you're going from DNA to RNA, uracil, U and A will base pair. All right, and then the third difference is that RNA is single-stranded. DNA is double-stranded, makes that double helix. RNA is single-stranded. So this picture from your lesson kind of shows the differences. We got, different double-stranded versus single-stranded, and the different nucleotides that make up each one. Now, there are also different types of RNA in your body. Like you just have the DNA in your body, but there's different types of RNA that do different things. So there's messenger RNA, and that one has the information on how to make the protein. Transfer RNA, that helps with protein synthesis. Ribosomal RNA also helps with protein synthesis. It makes ribosomes. And then there's also interference RNA that you hear about. It's not in this lesson, but you hear about it in the news and stuff because there's a lot of research around it. But interference RNA has to do with protein, regulating protein gene expression and protein production. Okay, so we're finally going to go over the first process. We got transcription. We're going from DNA to RNA. For transcription to happen, you need to have RNA polymerase, and this is the enzyme that builds RNA. And it happens in 3 steps: initiation, elongation, and termination. So initiation is starting, right? And transcription begins when RNA polymerase binds to the DNA. It binds at a region called a promoter. I don't think you need to know that at this point, but it binds to the promoter, and that's initiation. Elongation, the next step, is building that complementary sequence. So the polymerase will read one of the strands of the DNA. So the polymerase starts to unwind the DNA a little bit, and then it'll start making the complementary sequence. So again, it reads one, this is like zooming in into the RNA polymerase. It reads one of the strands, and it's building the complementary sequence. So when there's a T on the DNA, it's going to put an A on the RNA. When there's an A on the DNA, it's going to put a U, because we don't have T in RNA. When there's a G, it will put a C, the polymerase. And the polymerase is building the complementary strand. It's reading that DNA, it's building the complementary strand, and then that's elongation, so it's doing that all the way down. And then it's going to read a certain sequence in the DNA, and that sequence will tell the polymerase that it's done, and it falls off, and the whole process ends. And now you have your RNA transcript that can go do something. This is how mRNA is made. This is how tRNA, rRNA, all of those are made this way. Now I have a little practice here for you to practice coming up with the complementary sequence. So if this was your DNA, what would the RNA be? So go ahead and pause and write out what you think it is, and then hit resume and come check your answer. So here's the answer to the complementary strand, and there is a similar question on the quick check of coming up with the complementary strand. So remember, there's no T's in RNA, so whenever there's an A, the complement is U. All right, moving on to the next process, and this was a good time to pause if you need a little break. But next process is called translation. So now we're going from mRNA, specifically mRNA, to proteins. Now, proteins are made out of amino acids, so not nucleic acids, and amino acids also have a specific structure where they have an amine group, that NH. They also have a carboxyl group, the COOH. And then they have the side chain. The side chain is what determines the type of an amino acid, and there's 20 different amino acids. So there's 20 different amino acids that make all the proteins, and you put those amino acids in a certain sequence, and then if that protein folds up, it makes a 3-dimensional shape, and then that shape does something in the body. That thing will then go do something in the body, and the shape is important. Okay, so for translation, we need, first off, ribosomes. We learned about them previously. They're an organelle that make proteins, and ribosomes are made out of RNA. Now, on the mRNA, there are codons. So codons are 3 nucleotide sequences together, and they code for an amino acid. So we only have 4 nucleic acids that make up our RNA, but we have 20 different proteins. And scientists have figured out that 3 nucleotides in a row determine the type of an amino acid. And we have what is referred to as the genetic code. All right, and we put together this chart to organize all the different codons you could have. So all the different 3 combinations of nucleic acids you could have, and what the amino acid it codes for. So to read this chart, here's my pointer. So on the left-hand side, this is your first letter. So here we have U. Everything across begins with a U. Here we have A. Everything across begins with an A. Now up at the top, it tells us what the second letter is going to be. So in this row, everything in this row, the second letter is a C. So all of these have C as their second letter. All right, and then here's G. Everything has G as their second letter. And then on the third is or on this right-hand side is the third letter. All right, so looking here, the first letter is C. We go to this column, this box here, the second letter is C, so everything is CC. And then this is CCU, CCC, CCA, and CCG. Now there's a question about on the quick check about the code for serine. And the code for serine is AGU. So if we go to first letter A, second letter G, then we can see AGU. You'll notice that AGC is also an option for serine. There is redundancy in the code, right? There are multiple codons that code for the same amino acid. You can also see all 4 of these up here code for serine as well. You also hopefully notice that AUG is our start codon. Every protein starts with AUG. That tells the ribosome where to go, where to begin. And then we have 3 different stop codons. And when the ribosome gets to this codon, it stops. Okay, so translation. We have ribosomes, then we have the codons on the mRNA. The last thing we need to talk about is the tRNA. And the tRNA, the transfer RNA, carries amino acids to the ribosomes. And it brings the correct amino acid. And how it knows what amino acid to bring and to bring into the ribosome is with the anticodon. The tRNA has an anticodon. It is complementary to the codon. So the ribosome will only allow in the correct anticodon sequence and it will bring in the correct amino acid. You can see the tRNA here is carrying the amino acid at the top. So let's look at this. So in this picture, we have our mRNA, that's what's down here. We have our ribosome, that's what the bubbly thing around it is. And then these tRNAs here are inside the ribosome. We started with AUG, and you can see that the complementary, the anticodon on the tRNA was UAC, which are the complements. And you can see it's leaving the ribosome and it doesn't have an amino acid. That's what this circle ball is right here. Okay, it bound to this amino acid. And here's the codon. You can use the code to figure out what it's coding for. It doesn't tell us. But UUC was the codon, the 3 nucleotide sequence on the mRNA. And AAG is the anticodon, the complement. Our next codon, AAA, and then we have the anticodon UUU, and it's lysine. And then you can see the tRNA is coming over. That is the complement for this. So the ribosome is going to move down, and this guy will enter the ribosome, and then this one will exit. There are lots of videos that go through this process, and I highly recommend watching it. Okay, so then again, just to review, we're talking about gene expression, how proteins are made. So gene expression is this process of transcription and translation. Genes are segments of DNA that code for a protein, and the DNA is inside the nucleus. All right, the polymerase will read a strand of DNA, a template, and transcribe it to mRNA. And that mRNA can now leave the nucleus and go out into the cytoplasm to get translated. On the mRNA, we've got codons, which are 3 nucleotides that will code for an amino acid. So the ribosome will bind to this and translate it into a protein. Right? And again, each codon codes for an amino acid, These amino acids together will form a three-dimensional shape, and it will do something in the body. All right, last thing that gets brought up in this lesson, I know it's a lot, mutations. So a mutation is when there's a change in the DNA sequence. And so gene expression again goes DNA to mRNA and then goes to a protein. So if you change the DNA, you can ultimately change the protein and you can change its function. Now, you're going to learn more about mutations coming up, but let's just talk about hemophilia because there's a question on the quick check about it, and it's the one that's in the lesson that they talk about. Hemophilia is a genetic disorder caused by one DNA nucleotide being different in the DNA. So it's called a point mutation. One nucleotide was changed. That changes the amino acid that gets coded. And so then that changes one amino acid in this whole big amino acid changes protein, but it changes how it folds and it does not function well anymore. So shape does determine, the structure does determine function, getting back to our objective. So with hemophilia, people with hemophilia, their fibrin protein does not work well, and the fibrin protein is responsible for stopping any bleeding. So in this picture here, we've got a normal blood vessel across sections. We've got red blood cells and white blood cells are moving through. Normal blood vessel. But then sometimes it happens, you get a break. And that's, if you get a bruise, something broke a blood vessel, right? And you start to bleed. Now, your body has these platelets that have this fibrin in it that can release the fibrin and stuff, and it causes the break to be sealed and you stop bleeding. But people with hemophilia, the fibrin does not work well and does not stop that bleeding. And so a simple bang can cause a lot of bleeding and problems. All right, finally made it to the end. The multiple choice question. What processes make up gene expression? Transcription and translation, transcription only, or translation only? So what did we cover in this lesson basically? And if there's any questions, please reach out.